Hello my geniuses, welcome to another parasitology video today. So great science tutor Lorenzo, and today we'll be talking about the Entamoeba species. So we want to talk about all of them, the disease they cause, and how to diagnose treat them. So it will be important for your exams, and when you want to use it to diagnose and treat anybody, you could use my explanation from there. And if you have any question, you can drop it down in the comment section. And you also have to like and subscribe to my video, so anytime I'm dropping any video, you'll be sure to be notified. So you get it now. So if you want me to make any video for you, drop it down in the comment section. I promise I'll make the video when I say it. You get it now. So it's very important. So let us jump right into the video. So we are doing Entamoeba species. So Entamoeba. So we're talking about the Amoeba family here, because all this one we're talking about is protozoa. Yeah, and under protozoa you have the Amoeba there, you have the Euglena, you have the Paramecium, you have the Chlamydomonas, and we have talked about some of them and what they do and how they move. So this is the Amoeba. So we call it the Entamoeba species. Now the Entamoeba belong to a subclass called the Sarcodina. Hold it. It belongs to a subclass called what? Sarcodina, and a class called what? Rhizopoda. I get it now. So they are from a class called the Rhizopoda and a subclass called the Sarcodina. So Sarcodina is a superfamily. Yeah, so let's call it the family. So the Sarcodina is the family, while the class is called the what? Rhizopoda. So get it now. So how do they move? So your normal biology, they move to what we call pseudopodia or protoplasmic word flow. So they belong to a natural protozoa, but belong to a superclass called what? Sarcodina, and to a class called what? Rhizopoda. Yeah, now so they move to what we call pseudopodia or protoplasmic word flow. Some important parasites here are the Entamoeba, Iodamoeba, Endolimax, Dientamoeba. So you have the Entamoeba. These are the parasitic form of Entamoeba species that we'll be talking about. Yeah, now. So what is their nuclear features now? What do they have? Their nucleus, how it is now. So now they have what we call a vesicular nucleus. Their nucleus is just like in a vesicle shape. I get it now. So you also have small endosome near to the center. You have a small endosome which you are seeing there is much more centrally located. And peripherally, what do you have? You have what you call chromatin granules. Again, and so their nucleus is what? Vesicular in shape, very big, followed by they have a small endosome. And you get it now. You have peripheral chromatic granules. So that's how their nucleus is, because their nucleus is very important when you're talking about Amoeba species. Yeah, now, so we have talked about them, that they become to a superclass called Sarcodina, a class called Rhizopoda. They move to a what? Pseudopodia or protoplasmic flow. Yeah, now, and they have their vesicular nucleus, small endosome, you have peripheral chromatic granules. So now, what are the three? They are the species under Entamoeba. Now, what are the species there? One, you have the Entamoeba Histolytica, which is the main pathogenic parasite to human. So you have the Entamoeba Histolytica, you have the Entamoeba coli, you have the Endolimax Nana, and you have the Dientamoeba Fragilis. Okay, now, so don't forget, the one that causes issues to human is Entamoeba Histolytica. But you still have the one with coli, Endolimax Nana, and Dientamoeba Fragilis. Are you getting me now? So remember, now what is the Entamoeba Histolytica? Remember, the Entamoeba Histolytica usually affect the large intestine, just like the Bacterium coli. So most of them affect the large intestine, unlike Toxoplasmosis and Plasmodium. All those ones, they pass through lymph nodes, they destroy red blood cell and destroy various organs. But you see, the Bacterium coli and the Entamoeba species, especially the Histolytica, damages the wall of the large intestine. So epidemiology, now where can you see them? They are much more prevalent in the tropic and the subtropic areas. They are found everywhere around the world, not even cosmopolitan. They are found everywhere around the world. Children are much more susceptible because of their immune system, and also old people. And the reservoir is dog, pigs, and monkeys. Yeah. So what is the mode of transmission here? They are transmitted through contaminated food and water naturally. So when you eat contaminated food and water, you have more higher susceptibility to take any of the Entamoeba, especially Entamoeba Histolytica. Okay, so also all of them can be transmitted by that. Also, there's what we call Mosca domestica, which is your normal housefly you have in your house. So your normal housefly can also house Entamoeba Histolytica. So that's why you don't always open your food or water, because if fly drops on it, it will drop something there. And most of the time, it's the Entamoeba species, especially the Histolytica. Also, fecal to oral route from exposed to waste products that is already fecal, which can be transmitted from flies or maybe diffused to water, you drink it, problem. Also, you can take the cyst to meat. So when you eat a pork meat or a dog meat or a monkey meat, which you might not know, maybe you just eat as a meat and you eat, you ingest the cyst and start its life cycle in your body. So now, what are the parts? So how do you see it now? What is the life cycle? Apart from before the life cycle, you have to know different components of the Entamoeba Histolytica that causes diseases. So first one, we have the trophozoite, always know. We have the trophozoite. So the trophozoite here, you can watch it now. It's about 20 to 30 micrometer. Are you getting my length? Also, and in diameter also. It is roundish shape, is always roundish shape. And thin clear ectoplasm, which is its outer part. It has a thin clear, which is the membrane around. Thin clear what? Ectoplasm. And inside, which in the endoplasm, is granular, containing a lot of granules. It has a small central cryosome or endosome. So you can see the cryosome or endosome. So you can see also it has a fine what evenly distributed peripheral chromatin granules. So you can see the chromatin granules, which are the small white stuff inside there, inside the endosome. Also, the food vacuole may contain ingested what RBC. So you have the full barcode containing ingested RBC, because also they break down your red blood cell too, which can also cause anemia, which we'll talk about. So this is the trophozoite. The next one is the Pre-Cyst. What happens to the trophozoite? The trophozoite discharge undigested food, because after excistation, what happens? The excistation gives out what? Trophozoite. The trophozoite begins to multiply by time by binary fission. They become many, damage the intestinal mucosa, and thereby exist themselves and be released as feces. So first of all, before they exist themselves, what happens? They'll form what we call a Pre-Cyst. The Pre-Cyst is formed, not a complete cyst, just a Pre-Cyst that is rich in glycogen, and also the chromatin bar presentation. So it has a chromatin bar presentation, but the chromatin granules are not small, they are not round, they are in like a bar. So you see that yellow part is called the chromatin granules, but the chromatin granules is just like a bar shape, and they are rich in glycogen. It is not a full cyst, it's a Pre-Cyst. I get it now. So after it forms a full cyst, what happens? The full cyst now is spherical and oval. I will get it with about 10 to 20 micrometer. You get it now. So it's spherical over 10 to 20 micrometer, and it begins with one nucleus. I get it now. So it begins with one nucleus and matures to form about 4 nuclei. So it begins with one nucleus, matures to form what we call 4 nuclei. Also, they have tough hyaline cyst wall. Okay, now remember, spherical and oval, 10 to 20 micrometer. They begin with one nucleus and mature to form what we call 4 nuclei. So their cyst wall is lined by hyaline and it's very tough. And also a mature quadrinucleated cyst. So the 4 nuclei that is formed, they are much more what infective. Again, and so when they enter into the human, they become what? Effective. And when they enter, they start destroying the human. Again, so also there's what we call excystation now. So now we are from the cyst. The cyst is inside the feces. It was passed out. So when you ingest the cyst through feces or when you eat the meat, what happens? It enters into the intestine. It encysts itself. When it encysts itself, what happens? It starts forming trophozoites. So it begins the cycle again. So we have excistation, trophozoite, pre-cyst, and cyst. Excistation occurs, or you call it the metacyst breakout, forms what we call a trophozoite. The trophozoite latches on the wall of the intestine, damages the intestine, forms a pre-cyst that is rich in glycogen, and also chromatin bar granules are there and form a cyst. Okay, now. So if we see the life cycle here, so the life cycle here, you ingest the cyst which I've talked about by contaminated food or water. It enters into the intestine. It exists itself in the wall of the small intestine to form what we call trophozoites. So trophozoites multiply by transverse binary fission in the what? Large intestine. And what happens? Destroy the wall of the large intestine, also the small intestine, encysts itself in the colon and is passed out by what? Feces. Yeah. So now, what is the pathogenicity? So next one, what is the pathogenesis and the pathology? So just like the one we did in the Parasitology coli, the pathodiasis. So how does this affect the intestine? And also you see this one now, the erosion of the large intestine is much more deeper than the one we see in Parasitology coli. So what happens? The trophozoite possesses an active cysteine proteases. So in that one too, they release what we call proteolytic enzyme that start eating the wall of the mucosa. But this one, the trophozoites now releases what we call an active cysteine proteases. I get it now. So the lesion begins in the cecum and go to the appendix and start continuing the upper wall colon. And what happens? They start eating through the wall. Start with the epithelium, go to the lamina propria, go to the muscularis mucosa, and start going deep to the subwall mucosa. I get it now. Sometimes now when they destroy it, they become, first of all, it becomes ischemic, because if there's no blood supply to the epithelial wall or the submucosal wall, what happens? It will start becoming to dislodge and become necrotic. So becoming necrotic first because there's no blood supply, and begin to start dislodging. In other lesion, I get it now, the amoeba now will invade the submucosa, the muscularis externa, and also the serosa. So in Entamoeba Histolytica now, what happens? It will eat through, causing what we call perforation. But in the baladiasis and caused by Parasitology coli, it only eat up to the submucosa. So when there's old lesion, or when the infection go over and over again, it can eat through the wall of the large intestine, causing bowel perforation. Amoeboma now may form in chronic lesion. We call it amoeboma. So when there's often recurrent infection in that particular part, what happens? You may see it to cause many lesions, start perforating different wall of the large intestine. So you can see the intestinal lesion here. Look at the intestinal lesion. So the intestinal lesion is eating through. It started with the mucosa. It enters into the submucosa, where the exudation becomes shallow, begin to become necrose, and begin to dislodge. You get it now. Sometimes old lesion can go to the muscularis mucosa. Old lesion can go into the muscularis externa and the serosa. So it eats through. So extra-intestinal spread, unlike the Parasitology coli, it's just there inside the large intestine. But this one now, it can also spread. The trophozoite now can enter into the mesenteric venous. So when it enters it, damage up to the submucosa, and also damage the vessel. Inside the vessel, what happens? It can enter into the blood through the the mesenteric blood vessel. So we have the superior mesenteric and the inferior mesenteric, but here it's passing through the veins. So when it passes through the superior mesenteric venous, what happens? It travels to the portal system, to the liver. So remember what is forming the portal vein. The portal vein is formed by the superior mesenteric vein and the splenic vein. So when it passes through the mesenteric vein, what happens? It enters into the portal vein. From there, it starts going to the liver. In the liver, it damages the liver. So the liver is the first part it damages. So when it damages the liver, it forms a liver abscess there. So when it forms a liver abscess, you start seeing cirrhosis of the liver. The liver metabolism will reduce. There will be splenomegaly. Metabolism will reduce. There will be hepatomegaly. I get it now. So also it can spread through the lymphatic system to the lungs, where it will cause what you call pulmonary amoebiasis. I get it now. So because when there's high amount of Entamoeba Histolytica, it will cause what you call amoebiasis. So, but first of all, it is not called amoebiasis when it is just localized to the intestine. Yeah, now. So, but when it starts spreading, what happens? It will now form what we call pulmonary amoebiasis. So, enter into the brain too. I get me now. We call it the brain-eating bacteria too. Again, so it start eating the brain tissues too, which is also bad. So that is the secondary infection. So if it damages the intestine now, what happens is it causing diarrhea, like we know, abdominal cramps. I get it now. So also pain, malabsorption, you get it now, weight loss, nausea and vomiting, because you will not be able to keep those food down there, nausea and vomiting, general malaise, I get it now, headache, fever, because it will go to the brain due to presence of the trophozoite in the blood. The pyogenes will stimulate the brain, I get it now, through the PGE2, which will stimulate, increase the hypothalamic set point, causing fever, Headache too, due to increase in cerebral edema. Why? Because the CSF fluid will increase. The meninges will start stretching due to increase in CSF fluid there. Due to that stretching causes what? Headache. Also, there will be amoebic dysentery, whereby you'll be, when you are going to toilet, you'll be excreting blood. You get a bloody stool. Also, there will be abdominal pain. There will be hepatomegaly. There will be destruction of red blood, there will be destruction of RBC. So these are the clinical features you see here. So in severe disease now, I get it now, you'll be going to toilet about 15 to 20 stool per day. I get it, you're going to, like, severe diarrhea will occur. There will be peritonitis, whereby your bowel will be perforated. I get it now, there will be bowel perforation. I get it. Sometimes it may mimic what we call appendicitis, because if you infiltrate the appendix, it will damage the appendix too. I get it now. Also, there will be cardiac failure if you go to the cardiac muscle, causing what? Pericarditis, endocarditis, myocarditis, anyone. You get it now. So that is very, very important. So how do you diagnose them? How do you diagnose them? You take a stool sample. When you take a stool sample will be rich with the cyst. You see the amoeboid cyst there, which is the Entamoeba Histolytica. You see them in the cyst there. You get it now. So when you see them in the cyst, you take a wet mount, you stain them, you bring them out, carry out some experiment on them, stain them, put them on the slide. So in the slide there, you can do a wet mount or a fixed, or you do a stained smear. So when you stain it now, you either see a thick or a thin smear. So when you put it under a microscope, the cyst there, when you stain the cyst, what happens? You see that the natural feces will take one color, while the cyst will be highlighted more. So you put it inside the microscope and you see it. You can also use a zinc sulfate flotation in light infection. So if you put, also you take what you extracted from the feces too, or from the blood, you put inside zinc sulfate, you put it inside zinc sulfate solution, what happens? It will float due to light infection. You can also do a PCR, whereby you want to check the DNA of the antigen that is giving us the disease when you have infiltrated the blood. You can also do a monoclonal antibodies also to also get what you are looking for. You can also do an assay to an LSA, and you can also do an LFT to check an LFT, because if there's liver damage, what happens? The AST and ALT level will also rise. Okay now. So also you do a CSF too. There's what they call the brain-eating bacteria. So in the brain-eating bacteria also is caused by amoeba too. You can also see whether it's when it passes through the lymphatic system, it goes to the brain. So how can you treat it? Are you getting me? How can you treat it? So always remember what you call the MATI. The MATI is M-A-T-I. M-A-T-I. In MATI you have what you call metronidazole. So metronidazole is a preferable treatment to treat what? Amoeba species, the amoebiasis, any Entamoeba species that is causing infection. Also, you also have, just like you have the iodochlorohydroxyquinoline, so you can also use it. Also use tetracycline here. You can also use the arsenic acid derivative to treat. So you can use metronidazole, iodochlorohydroxyquinoline, I use tetracycline, and you can use the arsenic acid derivative. You call it MATI. So Entamoeba coli now, if I can look, the only one that causes disease majorly is the Entamoeba Histolytica. What about Entamoeba coli? Entamoeba coli, what happens? It is non-pathogenic to human colon. So when it enters into the colon, into our large intestine, it doesn't perform all those things Histolytica is doing. Yeah, now it feeds on bacteria, yeast, and other protozoa. So that is what it feeds on. I get it now. It has what we call an eccentric endosome. It has an eccentric endosome. It has a coarse peripheral chromatin, and when it matures, just like Entamoeba Histolytica, if it starts with one nucleus, and for Histolytica it forms what we call 4 nucleus, and that 4 nuclei are infective. I get it now. If it enters in the body, it causes infection. But this one, Entamoeba coli, what happens? They form what we call 8 nuclei, and the cyst is about 10 to 33 micrometer. The next is the Endolimax Nana. They are what? Small intestinal amoeba, which means they can also infect the small intestine. Only small. They are round trophozoites with reticulate cytoplasm. Yeah, now, which means when they encyst themselves, they give what? Trophozoite. But their cause for disease is very, very low. Unlike Entamoeba coli, Entamoeba coli doesn't affect the colon or the large intestine of the human. They only feed on bacteria, yeast, and other protozoa. But Endolimax Nana, they can also infect the intestine a little, and they also have a rounded or trophozoite. Yeah, now. So their pseudopodias are very short, and they have an irregular endosome. Why the Entamoeba coli has what? An eccentric endosome. There are 4 nucleus, so they start with 1 nucleus, and when they want to multiply by binary fission, they form what we call 4 nucleus. So they have 4 nuclei, just like the Entamoeba Histolytica. So Entamoeba Histolytica 4 nuclei, Endolimax Nana 4 nuclei. You get it now? So they are the same thing. That's why they can survive in the intestine of what? Human. Dientamoeba Fragilis. Dientamoeba Fragilis. Now, Dientamoeba Fragilis have 1 or 2 nuclei. They have the smallest nuclei. So their Dientamoeba Fragilis has 1 nuclei, while Endolimax Nana and Entamoeba Histolytica has 4 nuclei, while Entamoeba coli has what? 8 nuclei. Okay, now the trophozoites are round or what? Elongated. They have a delicate nuclear membrane. Are you getting me now? The food vacuole contains what? Bacteria. The food vacuole contains bacteria in this case. Unlike Entamoeba histolytica, whereby their food vacuole contains ingested RBCs. But for Dientamoeba fragilis, they contain what? Their food vacuole contains what? Bacteria. They are important diagnostic clue because their size, their diameter is very small, and they have 2 nuclei. You get it now? So they have no cyst shape. I get it. They don't have any cyst shape. So to compare them now, what will you say? Entamoeba histolytica is bad for human. Are you getting now? They are pathogenic to human because if they enter inside the body of human, they are very, very effective. They damage the body of human, but the rest do not affect human. So also Entamoeba histolytica and Endolimax Nana have 4 nuclei, 4 nuclei, while Entamoeba coli has 8 nuclei, while Dientamoeba fragilis has 1 to 2 nuclei. Okay, now. Also Entamoeba histolytica, their food vacuole contain red blood cells. Are you getting now? Why the Entamoeba coli, their food vacuole contains bacteria, yeast, and other protozoa. Why Endolimax Nana, their food vacuole contains some little stuff too. You get it now? Why Dientamoeba fragilis contains what? Bacteria. So I hope it all made sense to you and you have understood everything I've talked about on that Entamoeba species. It's very, very easy. I get them now. We say that they cause amoebiasis. I get them now. They cause amoebiasis, and they have the same life cycle like other protozoa infection, whereby they encyst themselves, they form a trophozoite. From a trophozoite, what happened? They multiply by binary fission. When they multiply, what happens? They destroy the intestine. They now encyst themselves and pass out towards the feces. So hope you understand. If you are new to my channel, make sure to subscribe and turn on your notification button so at any time I drop any video, be sure to notify. If you want me to make a video for you, drop it down in the comment section. I promise I'll make that video for you. So we'll be going to our next video, which is Trichomonas species. The Trichomonas species is also a protozoa species that causes diseases. So it causes trichomonasis. So we'll do it in our next video. So keep watching my video, keep subscribing to my video, keep watching it always, and share to all your friends and those sitting for the same exam. Bye bye.